Hey guys, so we're going to take a look at glucose and fructose in the 3D models. So here's the two of them, and we're going to start with glucose. So if you see here, you can count the 1, 2, 3, 4, 5, 6 carbons, but more importantly, you see the shape that's kind of like a hexagon here, which is different from fructose, which we're going to look at here now. Here's just that one for you. And what's different about this one is instead of there being 6 molecules here, there's 5, kind of like a pentagon. That really helps me. But there's still 1, 2, 3, 4, 5, 6 carbons. Now we're going to talk about deoxyribose and ribose. So if you want to come look at them next to each other and see how they're different. And what we're looking at here is how many oxygen there are on each molecule. So we're going to start with deoxyribose, which has the 1, 2, 3, 4 oxygen on it. And the way I remembered this one was it's the longer name, deoxyribose, but you'll see it's the smaller amount of oxygen. Or if you think like deoxyribose, the D, like it's a decreased amount of oxygen. So now we're gonna look at ribose, which you see will have 1, 2, 3, 4, 5 oxygen on it, and it's a smaller name, ribose, and there's more oxygen on it. So if you look at the two next to each other now that we kind of talked about that, you'll probably notice the difference a little more. Now we're going to take a look at maltose, which a good way to remember this one is there's multiple glucose, which is like maltose, you know. So if you come close, you'll see that maltose is a disaccharide, and we have 2 glucose molecules together. So I'll hold that up for you guys. You can see the 2 glucose, and if it helps, here's a single glucose, and you'll see how you can notice the shape there, how they're the same, and then as well. Oh. Finally, we're going to look at sucrose, which we know is a glucose and a fructose molecule together. So if you want to come and look at the model here while it's laying here, and if we look at it like this, you can see the glucose molecule here, 1, 2, 3, 4, 5, 6, and the fructose here, 1, 2, 3, 4, 5. And if you want to look at the molecules separately, we've got the glucose here and the fructose here, and then we will put them together, and then if we look at the big one again. Now we're going to talk about sucrose again, and how we have our glucose molecule here and our fructose molecule here. And in order to make sucrose, there's a little chemical reaction that happens called dehydration synthesis, and when that happens, we get a product of H2O. Mallory here is going to show us what happens. Nice. And then if we want to break it through hydrolysis, we would then gain the water back.
So here we have a triglyceride, tri meaning 3 fatty acids, 1, 2, 3. And if you take a look here, this is what a single fatty acid would look like. It's a long carbon-hydrogen chain with oxygen at the top. And along with the 3 fatty acids here on the triglyceride, we have a glycerol backbone, which here's what that looks like apart from that. So here we have a phospholipid, which has a phosphate group here at the top, and 2 fatty acids here at the bottom. These are found in the phospholipid bilayer of a cell. So now here we have a cholesterol, which has 1, 2, 3, 4 carbon rings. And we want to take a second to notice the difference between a fatty acid and a cholesterol, which you can notice with the fatty acid it's a lot skinnier, and you always want to count the rings with the cholesterol just to know the difference.
So now we're going to look at proteins. Proteins are made of a bunch of amino acids. There's about 20 amino acids, and right now we're only going to look at these 3 right here. Each amino acid is composed of the same groups. So each one has a nitrate group, which is going to be your blue, your carboxyl group, which is going to be the 2 oxygens and the carbon, your central carbon on those, and then each one is going to have a unique R group as well. Okay. Looking at each one, we're going to start with this one. This one's going to be your cysteine because it has the yellow one, which is going to be your sulfur. Next, we're going to go to glycine. It's the smallest one, and it has an oxygen on the central carbon. And then alanine has 3 carbons, so I remember that one because it's big Al, and it's bigger compared to glycine. Another way to compare these two is glycine, and alanine has alanine, looking at the syllables with them. You can take 2 amino acids with 2 amino acids. That will give you a dipeptide, which would look something like this, putting 2 together. You can also take amino acids, a bunch of them, to create a beta sheet, which would be something like this. Or you can take multiple amino acids to create a alpha helix, which would be something like this.
Next, we're going to talk about nucleic acids. Now, nucleic acids deal mainly with molecules that have nitrogen. So nitrogen is blue, and anytime you see a lot of blue in a structure, it's probably a nucleic acid. Now, this one specifically is a purine. Purine has carbons and nitrogen and 1 oxygen and 2 shapes to it. Now, purine is coupled with a pyrimidine, which is another molecule that has nitrogen, carbon, and oxygen, but it has 1 shape to it. So I always think the larger structure has the smaller name, purine, but the smaller structure has the larger word, pyrimidine. And these are both what are called nitrogenous bases. Now, if you take 2 nitrogenous bases, purine and pyrimidine, and you combine them, you're going to form a nitrogenous base pair, just like if you had a pair of aces in a game of poker. Now, they're held together, if you look very closely, by these white beads, which are the hydrogen bonds. So these are holding together the pyrimidine, which you can see here, 1 shape, and the purine, which has 2 shapes. Now, we can build nitrogenous base pairs on top of one another in a spiral-like motion or structure, and eventually you're gonna get...
Ding, ding, ding! Order up, Professor Klein! DNA!
Thank you. Just what I ordered. DNA. And as you can see, if we turn it this direction, we've got your purine, 2 shapes, and your pyrimidine, 1 shape, held together by the hydrogen bonds. Now, you're probably wondering, well, what holds the base pairs together on either end? Well, each base pair has what's called a sugar phosphate backbone that acts like glue or a staircase that holds in the nitrogenous base pairs. And you can see sugar phosphate. Here's a sugar phosphate group, sugar phosphate, and it will alternate between a sugar phosphate all the way down the outside of DNA. So zoom in here, you can see the sugar phosphate, sugar phosphate, as it continues up and around to make DNA. All right, we're here with one of my favorite molecules. This is ATP, or adenosine triphosphate. This one's got the purine-like structure up here, a sugar, but then the very distinct part about ATP is that it has 1, 2, 3 phosphates, which gives it its name triphosphates. So if you ever see 3 phosphate groups in a row, it's definitely ATP.